Hello and welcome to Simplified. My name is Gurjot
And my name is Michael.
and this is episode 1.
In this very first episode, we spoke to Michael Nash, an assistant professor of engineering of synthetic systems at the ETH Zurich and the University of Basel.
Which means,
Which means that, we expect him to be in the ETH Zurich, but we didn't realize that he was actually based in Basel.
yeah, and we realized it pretty late. We realized it a day before when we were actually supposed to do the interview. And in the end, we ended up doing the interview telephonically.
Now, the problem with a lot of the ETH buildings, as you remember, is that they're like a cage that absorbs phone calls.
So you don't get any cellular network inside pretty much all the buildings here at ETH. Well, man, that was such a pain. We had to sit outside in Zurich winter, minus 10 degrees, snow all around, middle of January, just the two of us sitting on a bench.
The plus side was, of course, that it was completely silent.
Yeah, that was the perfect recording environment, you could say, but we were shivering and sniffling our noses throughout the interview.
We spoke about nanomaterials, and what exactly was a technical term that...
Yeah, if I remember correctly, Professor Nash works with biomolecules and multicellular enzymes. He makes synthetic biomolecules and makes them do things they don't normally do.
Okay, so you mean basically Michael Nash messes up with life to create like awesome things like biofuels
Yeah, he creates artificial life that helps real life.
that helps real life.
Yeah, like one of the projects that he's working on is bioglue, you could call it, which is just glue that could replace ordinary stitches.
Oh yeah, so you mean like instead of stabbing people with needles?
Yeah, you just glue them up, right? And the good thing is they don't leave any scars behind.
Or maybe that's a bad thing because it's less badass.
Yeah, yeah, I'm still ambivalent about how I feel about this, but
Another thing he spoke about was biofuels.
yeah, biofuels they've gotten pretty famous recently. Actually, if you look at the energy debate, biofuels are fuels made out of crops.
Yeah.
That's basically what they are.
And
But
the
my
coolest,
favorite, yeah, the coolest,
The
and
coolest.
my favorite part was biopolice.
biopolice. That's right.
Totally scientific name we just made up.
So like in your body, there are the good guys and the bad guys. The good guys that help you live, and then the bad guys that want you dead. Well, so you've got this thing that Michael Nash has been working on, which is
Yeah.
biopolice.
Biopolice, yeah, again, this is totally scientific. These are just biomolecules that help identify the harmful bodies in your body, like, say, cancer cells. So you put these biomolecules inside the patient's body,
Yeah.
and they tag the locations where these where these cancer cells are located. They're like trackers, essentially.
And by tracking like the bad guys,
Yeah, you can
you're able
use
to.
cure people better. You can use targeted medicine because you know exactly where to hit.
Yeah, exactly.
And yeah, of course, Professor Nash explains this much better than what we just did.
So let's give it up for Michael Nash.
Professor Nash, thank you so much for joining us today.
Thanks for having me.
Welcome to Simplified.
Yeah, welcome to Simplified. So, straight up, what's the most interesting piece of research that you can tell us about that you do?
Oh, that's a very tough question to nail it down to just one thing, because I think a lot of what we're doing is pretty interesting, but we focus on the interface between synthetic and biological systems. And so we're trying to train biological systems to do things that they wouldn't normally like to do. And this can have then lots of different technology applications, industrial applications, also biomedical applications. And so this is what we do. We try to train biological systems to do synthetic activities that they are not normally trained to do.
That sounds good, but could you give us an example? Like, what kind of biological systems are you looking at?
Yeah, sure. So one of the probably best examples is stimuli responsive polymers and proteins. And so what we can do is take naturally occurring proteins and we look into their sequences, and then we find certain segments of these proteins that have this property that we want. Is called stimuli responsiveness, and this just means that it responds to the environment in a predictable way. So it could, for example, become insoluble or fall out of a liquid solution when you raise the temperature. And so we can use these types of proteins, and we build artificial versions of these proteins, and we can use them to do things like biosensing and drug delivery and biomaterials and these types of things. So this would be one example. So taking a natural protein, taking a portion of it, and then re-engineering it to be synthetic and artificial, so not naturally occurring. And then we can use this to build up systems for like some of those applications that I mentioned.
Wait, so you can create properties that you want out of these natural occurring substances?
Yeah, exactly. I can give you another example. One of the other examples we're looking at is systems that are involved in creating hydrogels. So we can use enzymes, which are not normally involved in the production of gels. Basically, you can think of a hydrogel like a jello. It's like a wiggly, jiggly little cross-linked, polymer material that has a lot of water. So it's swollen with water. It's a network of polymers swollen with water. And the way you create these...
I'm sorry.
Yeah.
Where do these biogels occur? Like, do you find them in animal bodies, or is it plant based? Where do you get them?
Find, so I'm talking about the synthetic gels, but of course they also do occur in nature. There are many proteins that actually naturally form gel-like materials. So, the extracellular matrix is a good example of a biopolymer cross-linked network that behaves like a gel. So this is just all of the space that's in the tissues that sits between the cells. So cells don't just sit there, in the body, in the tissue, completely isolated, but they're embedded in this extracellular matrix. And this is these fibrillar proteins. This is like a whole network of proteins, and that form a meshwork in which the cells live. And the interactions between the cells and the extracellular matrix is really important for signaling and for talking with the cells. And so this would be an example of a naturally occurring one. And then also
So cells, they kind of like, if you just, cells are like raisins stuck inside a jello. Is that a good analogy?
That is not such a bad analogy, yeah, but it's a little more interactive than just raisins, right? So you have the cells will sort of sense their little surroundings, and they can respond to chemical signals or to mechanical signals in the extracellular matrix. And this determines what we call cell fate. So it determines how a cell will behave and respond and how it will develop over time.
Okay.
Yeah.
So, so far, all your examples have kind of gone above our heads. So, is there something that we as non-technical people could possibly understand about biomolecules or polymers that you talked about?
Sure.
Like maybe an application in the pharmaceuticals or something.
Yeah, okay, sure. So I think one of the areas that we're looking at is, for example, in developing medical glue. So we just got an ERC grant, a starting grant to work in medical adhesives. So the challenge is to develop sort of a sprayable or injectable mixture of materials, of proteins and liquid dissolved in liquid, and these can form a cross-linked glue. And this glue will then be very biocompatible, non-toxic, and will stick very strongly to tissue. And it can be used, for example, in surgical procedures in order to basically replace sutures and stitches. And of course, medical glue is a big industry already. There's medical superglue, the cyanoacrylate-based superglues, and also there are fibrin gels. So there's a lot of technology out there, but we have an eye on studying the mechanical properties of gels and medical adhesives in a little bit of a different way. And so this would be one application area that we envision for this type of research.
So this glue that you talk about, I'm pretty sure it has benefits over the usual stitches or sutures that I used. So what could be the major benefit? Like, is it self dissolving, or
Yeah, so it's
what's the benefit
biodegradable, for
here?
example. It'll just degrade by itself over time. And I mean, a lot of these advantages are already being used in existing medical adhesives. But, advantages over sutures or staples are that you have lower rates of infection, you have better cosmesis, which means the scar tissue it looks not as bad, right? It looks better after the wound heals. And I mean, these are some of the reasons that people want to use glues. Also, they're quicker to administer than sutures in many cases. So you can just, apply a glue much quicker.
In a way, it makes you look less badass after you get your stitches,
Yeah,
because you say
you won't have those big
that,
knife wound scars. Yeah, to brag about to your friends. It's true.
yeah, so pirates won't be too happy about it, but
Yeah.
You've done some work on biofuels. What can you tell us about biofuels and how does it relate to multi-enzyme complexes?
Okay, so this research that you're referring to is again it's focused on building up artificial molecular systems, and so one of the things that companies are trying to do, it's already being commercialized, it's big industrial scale in some places now, is they're trying to take biomass, which means basically agricultural residue. This means like all the leftover stuff that you have when you grow, for example, let's say you grow corn and then you harvest the corn and then you're left over with the corn stouffer. So this is all the material that's usually just thrown away. Actually, they need to keep a portion of it on the field to replenish nutrients in the soil, but a good portion of it is just basically waste. And wouldn't it be nice if we could use this waste and convert it into energy? And specifically, we want to convert it into liquid fuel or other chemicals. I mean, it doesn't really matter. But the point is that what you can get from this agricultural waste is sugars. And so people are trying to figure out a way to make inexpensive sugar from all of the agricultural waste. One of the ways that you can do this is using multi-enzyme complexes, like you mentioned, and these are glycosyl hydrolase enzymes and complexes of enzymes, so many enzymes, which are just proteins, and they will cut these chemical bonds that are in biomass, that are in these plant cell walls. And then what you can do is, if you can develop a really efficient catalyst that can be very inexpensive, then you can convert this biomass into sugars and then use this sugar to power different types of chemical processes. For example, you can feed the sugar to engineered microorganisms. You can produce fuels this way. So the typical path would be to go through fermentation, right, to make ethanol. So basically brewing beer or producing ethanol from, by feeding the yeast these sugars that we get from biomass. So this would be a good way to go. But the problem with that is that right now ethanol is very cheap, and also in general petrochemicals are very cheap at the moment. Oil and gas and so forth. So I think the trend in the field is sort of looking for better products that you could produce this way. So stuff that is basically more expensive to produce using conventional means. If you could power a synthetic biology chemical plant to produce, for example, pharmaceutical compounds or other precursors for the chemical industry, and do this, basically from using trash that people would normally be throwing away, that this would be very economically feasible, realistic, and then potentially profitable. And so this is the research that we're also looking at. So we're looking at some of these most efficient degraders of cellulose, which are these multi-enzyme complexes derived from anaerobic bacteria called cellulosomes. So we can study the enzymes and study the mechanisms by which they bind together, they assemble. So we look at protein interactions that are responsible for assembling multi-enzyme complexes on the surface of bacteria. We study those interactions at single molecule level. We also do engineering of these interactions and a lot of protein chemistry, protein biochemistry to study these. And with the aim of basically developing improved tools for the industry.
Okay, you mentioned you can make sugars that could be used at what I think at the microscopic level, right?
Sure.
And you can also make this fancy beer that could be used as a fuel. So when you say fuel, do you mean as a replacement for petrochemicals, or only for microscopic processes?
No, no, no. For large scale, I mean, the idea is that you could scale this up and you could produce, thousands of liters of ethanol, for example. So ethanol is right now it's being used as a fuel additive, and so you could basically get these large quantities of fuel additives of ethanol from agricultural waste. So that has been already the trend. I mean, that is really established, I would say, by now, and it's being. I mean, I know there are pilot plants, for example, in southern Germany that are being, developed and launched right now to do this process on a large scale. But the economics behind this whole process is quite complicated, actually. It's really hard and challenging just to do from a chemical engineering standpoint, from the engineering of a whole process, a whole chemical process, starting from agricultural waste that sits on a field somewhere. How do you harvest that waste, transport it to a biofuel refinery? How do you, make the process as efficient as possible? You can't put much energy into the process, right? It needs to be net energy positive. So if it costs too much energy to do this whole process, then the whole equation falls apart, right? So you need to find an efficient way to do each step. So they're doing things like they're like so one of the components of the biomass that cannot be converted into sugars is called lignin. So one of the things that they're doing is, powering the plant, the burners through the lignin. So they can actually get heat from the lignin just by burning it. And this can be used to heat the whole process. And then the other question is, how do you actually produce these enzymes that are involved in chemical conversion? And, so all of these factors play into it. And the idea, the objective is to end up being net energy positive at the end, where you can get high quantities of ethanol out.
Okay, so how far do you think are we from just corn-based beer that you said that you could just put in your car?
So I mean, there's already been there's a lot of misinformation out there about biofuels. So a lot of times people sort of conflate first generation and second generation biofuels. So first generation biofuels were basically using crops that either produce starch that's very easy to break down into sugars. So that would be like corn-based ethanol, or using sugar cane, which is also very easy to harvest sugar from. You just crush the sugar cane and wash it with water, and then you get sugar. And so these were what we call first generation biofuels. And these got like a lot of bad press because basically it was this, conflict between using food crops for fuel. And this is really not sustainable in the long run. You don't want to be competing, you don't want food prices to compete with fuel prices. But the newer technologies that are coming online are not using edible parts of these plants. It's really all the stuff that we wouldn't be using otherwise. So like I said, all the waste. And these are the second generation processes, and these are much harder to do technically. And that's where the research comes in. That's where we try to develop improved catalysts and so forth. So that kind of
that sounds pretty
differentiates.
Cool. So even with the could you tell a little bit more about the bad press that you mentioned for first order fuels? Because doesn't fuel already kind of command how the food prices change, right? Because transportation and everything comes into account. I mean, in the bigger picture, shouldn't it make not so much of a difference?
Yeah, I mean, it's a good point. I mean, what the relationship is between fuel prices in general and food. But there, I'm definitely not the right person to be asking about these questions. It's way out of my expertise. So I know it's a continuing issue. But I mean, in the first generation biofuel debate, it was really a direct competition. I mean, either you can eat the corn or you can convert it into fuel. You know what I mean? It was like really very stark competition between the two. And
so like very Fox News style, you'd say.
Yeah, I don't think. I mean, it's pretty clear that that's not sustainable in the long term, and I also believe that it was probably mostly made sustainable based on a whole series of government subsidies and incentives that were in the U.S. These are basically subsidies to farmers to get credit for biofuels and also fuel subsidies for biofuels. so this is quite complicated in terms of the politics and the taxation and incentives that go behind all this. But I mean, the goal is to make it competitive economically without any intervention from, on behalf of the government. Wouldn't that be nice if we could do renewable fuels from waste,
Yeah.
Something that has like an intrinsic advantage.
Yeah, exactly.
As opposed to...
But like I said, I mean, it's not also clear whether or not, ethanol is going to be the best product for this process. So we can power, like we said, we can feed these sugars that we get to microorganisms. We can use microorganisms to produce many other compounds, not just ethanol. And so maybe there are other, chemical precursors or other chemicals that are going to be much more, have much higher added value by doing this process using, biological catalysis instead of conventional chemistry.
And
what do you mean instead of conventional chemistry?
So conventional chemistry meaning, this is like organic solvents, harsh solvents, and sort of organic synthesis. How you would make some of these, precursors in the chemical industry. So glycerol and other, many other chemical commodities that are used in huge scale. We could potentially replace some of those through a biological process.
And what would be some advantages of doing it through a biological process?
Would
Would it just be cheaper?
Yeah, potentially cheaper, also greener, right? So more environmentally friendly. I mean, none of this stuff requires organic solvents and waste disposal. I mean, this is all, let's say, aqueous, so water-based chemistry that the enzymes are doing.
so in the end, you can just dump your waste in the ground. It doesn't matter because it's green.
Yeah, it'd be green chemistry. I mean, it's really in this direction.
So since you're directly competing with the big oil industry lobby, don't you face any problems with that? Like, are they happy with this research going on?
Well, so I just want to emphasize again, I mean, it's not that, biofuels, it's sort of the end application of one of the projects that we're working on, but it's by no means like, the be all end all. It's more like these synthetic systems that we can produce, these biological systems. They have, potentially many applications. And so biofuels and enzymatic catalysis, this is one of the areas we're looking. But I wouldn't describe my research as being based on biofuels. That's not really the focus of the whole research. It is one of the problems and one of the applications that we're looking at. But in terms of competition with the oil and gas, I have really no comment on that at all. I mean, I've never run into any problem there or any. I have no interaction, no basis for that comparison.
Yeah, I mean that's all pretty interesting. So, what else could you possibly tell us? Since you said biofuels isn't the exact focus, so what other projects do you have running that could be interesting to know about?
So yeah, one of the projects I can tell you about is in this direction of basically diagnosis, so more of a biomedical application. And so what we can do is use antibodies, which are basically the body's natural defense against invaders. They're little protein molecules. They float around your bloodstream, and they sort of are like the police officers that sort of patrol your blood. And when they find an invader, they stick to it and they mark it for degradation. And so you can use recombinant antibodies, so artificial, so engineered antibodies, like non-natural, and you can basically raise them against any target that you want. So whatever we want to, whatever molecule we want our antibody to bind to, we are able to find these antibodies now. This is through directed evolution. And so if you want to, for example, develop an antibody against a cancer marker, you can do this, and this has been done, of course. And now what we do is we can attach what are called photoinitiators. So these are molecules that can be linked to the antibody. And then when the antibody sticks to a cell, a target cell, a cancer cell, then what we can do is irradiate it with light. And then this photoinitiator will create a polymer, and the polymer will stick to the cancer cells and encapsulate the cells.
Wait, so you're not just tagging it, you're also trapping it in some way.
Yeah, exactly. And then the idea is that what you can do is if you have these very rare cells that are, for example, floating in the bloodstream, and you can capture them, encapsulate them in a polymer, in a gel, and then you can actually protect them against a lysis buffer. So this is, for example, surfactant, or that will actually basically pop open all of the other cells in the mixture, but your cancer cells that have been protected by the gel will remain healthy inside this protected cage. And the reason you'd want to do that is because you want to isolate these rare cancer cells, and then you want to study them, or you want to count them, for example, see how many there are. So you can sort of play these games where you protect certain cell types, and then you select and screen for them, and then you can study them individually.
Okay, so it's very targeted antibodies that you can generate. So that sounds very cool. And because what I was wondering was, can you also help patients who have a not so strong immune mechanism of their own? Can you just inject these antibodies into their systems and boost their immune system?
Well, introducing foreign antibodies, I think, is a little bit tricky in terms of boosting the immune system. I mean, I think it's more along the lines of cell therapy that can happen. If you just take an external antibody, it'll be recognized by the patient as being foreign, right? But what people can do is, for example, they can harvest immune cells from a patient and then basically train the patient's own cells to produce specific antibodies. But again, this is not really my area of expertise in terms of the cell therapies, but I know there is definitely research being done in this direction. I mean, this platform that I'm talking about for doing encapsulation of cells in synthetic hydrogels, this is more of a diagnosis or a biosensing kind of. So we would take a sample outside of the patient, and then we could, encapsulate a certain cell type using these photoinitiators, and then detect them, isolate them, study them, and so forth.
Okay, yeah, that makes sense.
Not for in the body necessarily, though.
Okay.
Oh, so you'd take out a sample and then include these.
Yeah, I mean, like
antibodies.
a blood sample is what we're thinking right now, that you'd have these fluid biopsies. So, I mean, even if you have a solid tumor, for example, there's a very small number of cancer cells that are then, basically sloughed off of the tumor and are circulating in the bloodstream, and these can lead to the metastasis of the cancer, of the tumor, to other parts of the body. And so, if you can screen and detect these very, very rare cells in the blood, then you can get a head start on that and detect it earlier.
Yeah, so how could this really change the game in terms of diagnosis for diseases?
Can't you already kind of recognize where cancerous cells are? Like, does this provide, like, what's the advantage of doing this?
Yeah, I think the advantage is that you can. I mean, the problem is the background. So if you're fishing for these cells in an ocean of other cells, then they're very hard to find, and so there's a limitation in terms of the number of cells that you can screen, and so you have this limitation in the technique that's used for this is called flow cytometry, and usually you're screening about 10 to the 7 cells per hour. I think that's a relatively optimistic estimate for the number of cells. So if you have a lot lower cell count than that for these rare tumor cells that you're looking for, then basically you'll never find them in this background of cells. And so by protecting the cells that you're looking for through this hydrogel reaction, and then killing and lysing all of the other cells in liquid, then you're basically lowering your background. So you're just increasing your chances that you actually find the needle in the haystack that you're looking for.
Okay, yeah.
Wow.
That makes
And
sense.
this would make it cheaper and faster to diagnose.
Yeah, I mean that's the end goal. That's the ultimate goal. I mean, I should also make a point that I'm not the only one working on this. I mean, there's many other talented groups, all over the place. So in Japan and the U.S. and Europe, who are also working on these similar types of cell encapsulation reactions. But this is one of the directions the field is moving.
Okay, yeah, this was all pretty cool, but now I think that's enough of science. But before we end, what resources would you recommend for someone who's interested in learning more about, say, these multi-enzyme mechanisms or biofuels in general? Off the top of your head, what do you guess is a good resource to read more about, apart from Wikipedia, of course?
Let me think. I think, well, honestly, if there's somebody who thinks they might be interested in getting into this in terms of research, like a bachelor student or something, my number one recommendation is to just get into the lab as soon as you can. So volunteer if you can, or whatever you can do to just join any research group that is doing something similar or something that you think you might be interested in, and just getting some hands-on experience as soon as possible. Because the earlier you get your hands a little like dirty in the lab, so to say, the better you're. I mean, number one, you just the learning curve is sort of sigmoidal, I would say. So you need to get on that low productivity portion, an early portion of the curve, so that you can get to the exponential growth as fast as possible. But I mean, that's the number one thing. Like, I think if people go into a research group, they maybe have some expectations, but I think once you see how it's really done, all of your expectations will completely change. So you really don't know what it's like until you see it firsthand and you interact and you do these activities yourself. And I think that's a real turning point for people. And it's one of these times in a student's development where it's just so exciting, and because everything is so new. It's all so new, and you sort of have no idea about what is all possible. But then when you start seeing this stuff firsthand or seeing how this stuff is done, it's just a really, really great and stimulating experience I would really recommend. So, I mean, I guess I don't know who's listening to your podcast, but if these are students at the ETH, for example, there's obviously many, many like world-class groups. And, just volunteer washing dishes for a few hours a week if you can, just to start with, whatever you can do to just get your foot in the door and start interacting with these amazing scientists that are all involved at ETH. If somebody's in Basel listening, and you're interested in this, come see me. We're happy to have you wash dishes or whatever, even just sit in on our group meetings or whatever it's going to take, because it's such a great opportunity, I think. But I mean, also for listeners, anywhere who are involved in a university setting, that they can just get involved and try to start learning some of this stuff. I mean, beyond that, I would say there's a lot of good scientific articles, so you can start with review articles, for example. So there are these journal articles that are reviewing a field of literature, and these are really helpful to get into something for the first time.
Could you name something for your field? Like, what would be a good journal to look at?
So any of the top journals in the field, so, Nano Letters, Nature Nanotechnology, Journal of American Chemical Society, Angewandte Chemie, these are all, good journals, ACS Nano, these are all some of the top journals in the field, and they're definitely publishing review articles as well in some of those journals, so you can start there.
Okay. One more question. What does your Bookmarks bar look like?
I have a very sparse digital environment in which I work. So actually, I believe. Hold on, I'll bring it up right now. So my Bookmarks bar, it has four bookmarks. I have.
That's it.
Paper.
Wow.
Sorry, no, it's five. It's five here. I have a website called Paperpile, which is what I use for scientific literature. So it's just basically a PDF manager that lets me read and cite science literature. Then I have Google Docs. So I do all my my scientific writing in Google Docs, and that lets me share my articles with my team members so we can work on them together. And then I have a link to my lab homepage, and then I have a link to our internal lab homepage, which is where we keep all the Secrets. Internal materials and stuff to organize the group. And then my last link, which is maybe the only interesting one, is a color schemes generator. So it's a website that will generate color schemes. So whenever I prepare graphics, I don't just randomly pick colors. I usually just generate a color scheme at this website, and then all the colors are like complementary and they match and they look nice together. So that's all that I have on my bookmark bar at the moment. Yeah.
So what programming language do you use for your work?
We use actually a few different ones, but one of them is called Python. That's one of the ones we're using to do some of the analysis. We also use Igor Pro, which is its own software program with its own special syntax and language. And then all the sort of more typical ones. I mean, all the standard, commercial packages, Microsoft Office, and all this stuff.
Okay. Since you're also a Professor Nash, are you ever confused with John Nash, the economist?
Sometimes I get comments about this. I've never been confused with him, certainly not. But usually I'll get some questions about if we're related, but there's actually lots of Nashes. So there's Robbie Nash, he's a surfer, and there's like Crosby, Stills, and Nash, like the country music group. So it also depends on the age of the person who I'm talking with, and so maybe some older people have a different Nash in mind. It's not an uncommon name in the U.S. at all, but actually in Germany and in Switzerland, it's very hard for people to spell this correctly. So if I say the name, then they don't really, because it's not a German spelling, right, of the Nash. So I get Nasch a lot,
Oh, yeah, that makes sense.
Kind of like Nash, like Naschen. And actually the nickname, so the group in Munich actually sort of, it all goes back to one colleague of mine named Stefan Heuke, and he ended up coming up with the nickname for my team called the Naschkatzen, which in German is somebody with a sweet tooth is called a Naschkatze, and then this got morphed into the Nash cats in English. Yeah.
Okay. So we'll keep an eye out for the Nash cats if you ever come across.
Yeah. So thanks to Stefan Heuke for that. I should give him a shout out. He was the original inventor of the Nash cats.
That's all very interesting. Thank you so much for giving us your time.
This was an amazing interview. So we're very glad to have talked to you.
Yes.
Great.
And we hope.
It was a pleasure.
Yeah, we hope you had a good time teaching us about these things.
Thanks a lot. Thanks for having me.
Thank you for listening. If you liked it, we'd really appreciate it if you shared it and gave us your feedback. We publish a new episode every week. For more details, visit our website simplified.xyz.